Good day, everyone. Today we would like to continue to Golgi apparatus. So, I think you already know what is Golgi apparatus. So, this is a Golgi apparatus. Golgi apparatus consists of a flattened membranous sac called cisternae. Each of these are called cisternae, and small fluid-filled membrane enclosed vesicles at the end. You can see here the membrane enclosed, or the called membrane bound organelle vesicles. The cisternae appears to be lined together like a stack of saucers, see, like this one. Then the Golgi apparatus appears to have three functional distinct parts. One is called the bottom, middle, and the top. So the bottom part we call is known as the cis phase. Okay, cis phase and the nearest to the nucleus. So the middle is at the middle regions, whilst the top is known as the trans phase that lies closest to the surface of the cell membrane. These three parts of the Golgi apparatus contain different enzymes and perform different functions. So like this one, this here is a cis phase that near to the nucleus. So where here is the, like for example, the nucleus, and then they have the ER. So the RER, and then they have the transport vesicle. So the transport vesicles will fuse to the Golgi apparatus. So this is the cis phase. Then here is the trans phase, where they're near to the plasma membrane and the place where the secretory vesicles are formed. Okay, so like here you can see, here's the nucleus, and then this part is the cis phase here. So cis phase, and then here is the trans phase, near to the plasma membrane. So the Golgi apparatus has several roles. First, receiving proteins from the ER. Here ER refers to the RER and SER, and may further process and modify them through the glycosylations. Glycosylation is this one, they add the carbohydrates to the polypeptide. Phosphorylation, the additions of the phosphate group into the polypeptide. And also proteolysis, that means to cut the polypeptide into the several fragments. Then the second function is to concentrate and sort and packages protein before they are sent to the cellular or extracellular destinations. Then synthesize the mucus for animal cells, extracellular polysaccharides, for example, the glycoproteins. Okay, glycosylation, as you know, is the addition of the carbohydrate to polypeptide. So it's essential for the addressing proteins to lysosome. So that means if the polypeptide they synthesizes will be modified to lysosome, so this glycosylation is the compulsory process. And some recognition function at the surface, because glycosylation they add the carbohydrates to the polypeptide, so then becomes what we call the glycoproteins. So this will be the antigens. They add as antigen for cell recognitions. And then also it's used to stabilize the proteins stored in the storage vacuole. Phosphorylation is the addition of phosphate group. So the phosphates come from the ATP. So that's why they use a protein kinase to hydrolyze the ATP into ADP and inorganic phosphate. So the phosphate group will be added to the polypeptide. So proteolysis is the cutting of the polypeptide chains. So the cleavage of the signal sequence from the growing polypeptide chains in the ER and the Golgi apparatus. So these three processes we can categorize as modifications and processings of the polypeptide that received from rough endoplasmic reticulums. So in general, the Golgi apparatus acts as the cell's post office. Okay, it's like receiving, then the sorting is chemically modified. Okay, and delivering the proteins and lipid for internal and external users. So remember the regions, okay, the regions that nearest to the nucleus, okay, is known as the cis phase. Then the protein modification and processes occur in the Golgi apparatus. So the process such as glycosylations, proteolysis, and the phosphorylations. Then after that, the final products will be packed into the secretory vesicles and then to be excreted out of the cell, or they'll be formed for the cell intracellular use. Okay, the Golgi apparatus receives proteins from the rough ER. So as I told you, as a cis phase here, then it packages them. Then the proteins from ER is safely enclosed in the transport vesicles to Golgi apparatus. Then also receive lipid-filled vesicles that are from the smooth ER. Okay, so remember, not only receive the protein from the rough ER, they also receive the lipids that synthesize from the smooth ER. Okay, remember this. Then once it's arrived in the Golgi apparatus, so the vesicle fills with the cis phase of the Golgi apparatus and releases the contents, either proteins or lipids, into the Golgi apparatus for processing and modifications. Then other small vesicles may move and transporting protein between the cisternae, they mean from one cisternae to another cisternae for other process or reactions. Then the vesicles budding off. We use a budding off or bud off. Okay, from the trans phase. So this is here is a trans phase. So from the trans phase, then they're formed into the either is lysosome for the intracellular use, or be packed into the secretory vesicles for extracellular purposes. All right, so this is the formations of milk proteins, okay, and the lipids in the cells. Okay, like the milk protein genes transcribed to mRNA. So here's the DNA. Let's say here there is a genes, okay, in the DNA which is used to transcribe the milk protein. So the information from DNA is copied, or we call it transcribed, into an mRNA. So then this mRNA travel or diffuse out of the nucleus and then attach to the ribosome. So in the ribosome, the proteins, or we call it polypeptide, is synthesized by translation. Okay, then the polypeptide they form is inserted into the lumen of the ER. So actually in the lumen of the ER, there is a modification of proteins occur or take place, especially the glycosylations. Then after that, they are brought off. Okay, they are brought off into the transport vesicle and then to be transported to the cis phase of the Golgi apparatus. Then in the Golgi apparatus, the proteins is processed and also modified, okay, through glycosylation, proteolysis, and phosphorylation, and so on. Then after that, the final product, which is the milk proteins, will be packed into the secretory vesicles and then to be excreted or secreted out of the cell. Okay, so this is how the proteins, okay, is produced. Okay, from the starting from the nucleus and then to the plasma membrane. So here, this is known as the endomembrane system. Okay, remember what I explained to you, what is the endomembrane system. Okay, some blood vesicles, they form lysosome. This one for intracellular use. Okay, like this one. Okay, they burn off as a lysosome, or we call a primary lysosome. So Golgi apparatus is abundant in rapidly dividing cells and secretory cells. But the cells that are for the secretion purpose, okay, and they are actively in cell division. So they have a lot of abundance in the Golgi apparatus. So for example, pancreatic cells, goblet cells, gland cells, and also reproductive cells. So these 1, 2, 3 are for the as secretory cells. So this one is actively for the cell divisions. Okay, for example, the pancreas secrete digestive enzymes. Okay, goblet cells secrete mucus. Okay, plant cells they need to produce the cellulose. Okay, the cellulose is produced from the what we call from the Golgi apparatus. Okay, during the cell divisions. Okay, so this is the process. You see, as a summary. So you must remember anything the proteins that they want to produce or synthesize must come from the nucleus first, because the nucleus they contain the DNA in which the genetic information is stored. Okay, then the genetic information will be copied into the mRNA, and then to be what we call a travel out of the nucleus, attached to the ribosome where the polypeptide is synthesized. Then after that, the polypeptide will be what we call transported to the Golgi apparatus for further processes and modifications. Then later, the proteins will be excreted or will be secreted out of the cell, or as lysosome for intracellular use. Okay, so that's all for the Golgi apparatus, and then thank you for the listening.